Hi everyone, today we are going to be solving Cambridge International ASN A-Level Biology 9700 Paper 51 May-June 2023. Catalase is an enzyme that is found in many plants and animal cells. Catalase breaks down hydrogen peroxide, which is a waste product from cell metabolism. Figure 1.1 shows the breakdown of hydrogen peroxide by catalase. So we can see hydrogen peroxide broken down by catalase to produce water and oxygen. A student decided to investigate how catalase activity varies along the length of a carrot root. Figure 1.2 shows a photograph of some carrots. We can see the carrot root, the shoot, and the leaves are labeled. The student extracted catalase from different parts of the carrot tissue. Carrot tissue was mixed with a same volume of pH 7 buffer and some fine sand. So the sand will work as a form of, so that it can be smashed nicely, The mixture was ground with a pestle and mortar. The mixture was placed into centrifuge tube and spun in a variable speed centrifuge for few minutes. This caused the mixture to be separated into a solid pellet at the bottom of the centrifuge tube and a liquid carrot extract at the top as shown in figure 1.3. The liquid carrot extract was poured into a clean test tube. This liquid contained catalase. Figure 1.3 shows a centrifuge and centrifuge tube with its content after spinning in the centrifuge for a few minutes. We can see the lid of a centrifuge tube with the plastic centrifuge tube of 2 cm cube. The liquid carrot extract was extracted at the top and the solid pellet which was thrown away. State one variable that should be standardized in the procedure to obtain the liquid carrot extract other than the carrot tissue, fine sand and apparatus that is used. Because in this particular process, if you notice, we are using a buffer. So they said small volume. Instead, we need a fixed volume for each extraction. Then we are using a centrifuge. Since we are using a centrifuge speeds at different, spins at different speed. So the speed of the centrifuge. Then obviously the centrifuge in terms of spinning it has different length of time that we can spin it for. So the time for this centrifuge spinning. So we can write any one of the 3 points as our answer. So the answers can be the volume of pH 7 buffer or the speed of the centrifuge or the time in centrifuge. This can be standardized. So any one of the points will be our correct answer. So just choose one of the point and write it as your answer. A student carried out these steps to investigate the activity of catalase in the different samples of a carrot root tissue. A disc of filter paper of 5 mm diameter was dipped into the liquid carrot extract using forceps. The filter paper disc was placed at the bottom of the beaker of 1% hydrogen peroxide solution using forceps. The time taken for the filter paper disc to rise to the surface of the hydrogen peroxide solution was measured. So when we are taking a 5 mm diameter disc and we are dipping it into the liquid extract, the liquid extract consists of catalase enzyme. Then we are placing it in a 1% hydrogen peroxide solution. So by the action of the catalase enzyme with the peroxide, we are releasing oxygen. So let's say this is the paper disc. As the oxygens are forming, they're forming bubbles all around it, and this will cause the paper disc to rise on top in the liquid. It will rise on top because the disc gets lighter as the oxygen bubbles are produced. Figure 1.4 shows the apparatus used to measure the time taken for the filter paper to rise. So then we are measuring what time it takes in seconds for the filter disc to rise. We are using a 10 cm cube beaker and a 5 mm filter paper disc with 1% hydrogen peroxide solution. State the relationship between the time taken for the filter paper disc to rise to the surface of the hydrogen peroxide solution and the catalase activity of the liquid carrot extract. So since the different place of the carrot is used and the catalase that is extracted, they are going to be the same catalase but different concentration. So it will give a relationship like this: high concentration. So technically it should give high concentration is equals to faster rise to the surface. Now, so we can write the answer in a way: the time for the disc rising decreases as catalase activity increases. The student decided to investigate how catalase activity varies along the length of the carrot root. So a carrot is measured and we can see increasing distance from the base of shoot. The student made the prediction: as the distance from the base of the shoot increases, catalase activity of carrot root tissue increases. So according to the student, there are more catalase at the bottom and less catalase at the top. Identify the independent variable in this investigation. So the independent variable is, since we are measuring the distance and we are going to choose where we are going to collect that piece of carrot tissue to use in our experiment. So we can say distance from the base of shoot, because this is the point that we are actively choosing. Describe a method that the student could use to test the prediction that as the distance from the base of the shoot increases, the catalase activity of the carrot root tissue increases. Your method should set out in a logical order and be detailed enough to allow another person to follow it. Details of how to use the centrifuge to prepare the liquid carrot extract should not be included. So this part is excluded because this was mentioned before. Now we would have to write a method. So the method would be the procedure, the process. The process we can use to test the prediction. The process of testing our assumption or prediction. Okay, now we are not going to include how the liquid extract is produced. So what we need to do, we need to set it in a logical order and detailed enough for another person to follow. So we can mention as instructions, similar to the one that we have received in our paper 3 during our practical. Cut tissues from the carrot root at 3 cm/2 cm interval. Start at 0 cm, 2 cm, 6 cm, 8 cm and 10 cm and at 12 cm depth. Use 1 gram of cuttings from each depth to make the extract. Maintain the temperature of hydrogen peroxide solution throughout the experiment at 25 degrees Celsius using water bath. Use hydrogen peroxide of 5 cm depth for all experiment. Mix liquid carrot extract and then dip the filtered paper disc into the carrot extract. Measure the time taken for the extract soaked filter paper to rise to the top from the bottom. For every distance of shoot, repeat the procedure with 3 filter paper discs and calculate the average/mean time taken for the paper disc to rise. Now 2 added points, write any one of them. One is for safety: wear gloves, eye protection to avoid contact with hydrogen peroxide, which is a hazard. Now other than that, you can say perform a control experiment with disc dipped in water/buffer only to show that the rise of disc is due to extract only. In a different investigation, the student determined the effect of hydrogen peroxide concentration on the activity of catalase in yeast. 5 cm³ of hydrogen peroxide solution was placed in test tube at 25 degrees Celsius. 0.2 gram dried yeast was added to the test tube, so the yeast cell formed a suspension in the hydrogen peroxide solution. A gas syringe was quickly connected to the test tube using a bung and delivery tube as shown in figure 1.6. The volume of gas collected after 1 minute was measured. So we are doing a different investigation where we are determining the effect of hydrogen peroxide concentration. So that the variable that we are changing in this one, the independent variable, is hydrogen peroxide concentration. Now to do the experiment, we are keeping few things constant, which is 5 cm³ hydrogen peroxide solution. So we are keeping the volume of hydrogen peroxide constant. We are placing it at a test tube of 25 degrees Celsius. So we are keeping temperature constant. And we are adding 0.2 gram of dried yeast. We are adding 2 grams of dried yeast to the test tube. So we are keeping this is the addition of the dried yeast. This is also a constant variable. Mass of yeast is constant. Now we're using a gas syringe, alright, was quickly connected to the test tube using a bung and delivery tube as shown in figure 1.6. The volume of gas collected after 1 minute was measured. So the volume of gas that is collected will be our independent variable. Figure 1.6 shows the apparatus used by the student. So we are seeing a gas syringe connected with the delivery tube and rubber bung to seal the gases so that they cannot escape, and bubbles of gas being produced due to the yeast addition in the hydrogen peroxide solution. Identify the dependent variable in the investigation. So the dependent variable should be the volume of gas that is collected. So the volume of gas collected after 1 minute is the dependent variable. To investigate the effect of hydrogen peroxide concentration on the activity of catalase in the yeast, the student first prepared a range of known concentration of hydrogen peroxide. Now, we are preparing a range of known concentration of hydrogen peroxide because our independent variable is the concentration of H2O2, the hydrogen peroxide. Now, the student was given 6% stock solution of hydrogen peroxide. The student decided to use the stock solution to make hydrogen peroxide solutions with concentrations of 3%, 3.6%, 4.8%, and 5.4%. The student made 50 cm³ of each solution. Complete the table 1 to show how this solution could be made by proportional dilution of 6% stock solution of hydrogen peroxide. Now, percentage concentration that we are producing is given here. We can use the formula V1 times C1 is equals to V2 times C2, where C represents the concentration and V represents the volume. So, in the first, we don't know how much volume, initial volume of 6% we will have to add. So let's do one of them for the 3% hydrogen peroxide concentration. So we don't know how much V1 we will have to add, which is the stock. So V1 is stock solution. However, we know the starting concentration, which is 6. Now we will have to make it into a volume of 50 and times the new concentration, which is 3. So the requirement for the V1 will be 50 times 3 over 6. That gives us 25. So we will require 25 cm³ of the stock solution. Now to make it into concentration of 3%, we will have to add water to it. So we will mention the volume of distilled water that is added. So here we are going to write distilled water slash cm³. So volume of distilled water slash cm³ will be required 25 cm³. Now we'll apply the same maths for all of them. So each and every time the math is going to be like this: 50 times the concentration that we want to get it into divided by the concentration we're starting from. So we'll require 30 here and 20 cm³ of water. So we were given the space below for working. However, since we have done the working within the table, just make sure that you don't do the working within the table. You rather mention the working below. Figure 1.7 shows the results of the investigation. Volume of gas collected after 1 minute, percentage concentration of hydrogen peroxide that we have used. So it started from 3.0 and we used up until 6.0, and this was the volume of oxygen gas that was collected in each instances. The student made two conclusions. As the concentration of hydrogen peroxide increases, the volume of oxygen produced after 1 minute increases. Vmax of catalase is 88 cm³. And second one is Vmax of catalase is 88 cm³. Now Vmax of catalase cannot be determined by looking into this experiment because the line didn't plateau after a certain concentration of hydrogen peroxide. The line must plateau in order to find Vmax. Use the information shown in figure 1.7 to evaluate this conclusion. So first conclusion is, as the concentration of hydrogen peroxide increases, volume of oxygen produced after 1 minute increases. So the data that we see from the graph, it supports our conclusion. So we're going to write at 3.0, when we extrapolate, the volume of oxygen gas collected was 37 cm³. And at 6.0, the volume of oxygen collected was 88. So we will use a comparative data code. So at 3%, the volume of oxygen collected is 37 cm³, and at 6%, volume of O2 collected is 88 cm³ in 1 minute. This supports the first conclusion. Now the second statement is not supported. However, the second statement is not supported because the range of hydrogen peroxide is limited in the experiment, and finally the graph has not plateaued at the end, so we do not know the value of Vmax. Sea trout. Salmotruta are found in the rivers and coastal seawaters in many regions of the world. Sea trout lay their eggs in rivers and the young fish remain in this freshwater environment. After living for 1 to 8 years in the freshwater rivers, sea trout migrate to the sea. Adult sea trout feed on coastal seawater regions, usually no more than 80 km from home river. Fish farms that produce Atlantic salmon, Salmon salar, may be located in the coastal seawater regions where wild sea trout are also found. Figure 2.1 shows one cage at a fish farm. So we can see no nets to stop the birds on top, and then the fish cage where the farmed Atlantic salmons are kept and wild sea trouts are everywhere outside the net. The increase of fish farming is thought to have caused more infection by salmon lice, Lepiotherius salmonis, in wild fish such as sea trout. When sea trout enter coastal seawater regions, they can become infected with salmon lice. Salmon lice larvae live in seawater and can move freely between fish cages and open seawater. Salmon lice attached to the skin of the sea trout feed on their skin and the blood. The resulting tissue damage can sometimes cause the death of the sea trout. In figure 2.2, a sea trout infected with salmon lice can be seen. So the salmon lice are attached to the site in the body of the sea trout. Some scientists found out from a published source that 13 or more attached salmon lice usually cause the death of a sea trout kept in a laboratory condition. The scientists decided that each wild sea trout with 13 or more salmon lice is at risk of physiological stress and potential death. Outline a laboratory method that could be used to determine the number of salmon lice needed to cause the death of sea trout. Suggest a suitable control that could be used to confirm that it is the salmon lice that are causing the death of the sea trout. So, first of all, let's talk about the method that could be used. So, the method that could be used is by adding sea lice to container and counting the sea lice on the trout at death. And a suitable control would be repeating with container of trout with no sea lice that is present on that particular container. Now, to keep the experiment fair, the temperature of the water should be kept constant in both experiment and also the age of the sea trout. The scientists investigated the relationship between fish farming and infections by salmon lice on wild sea trout in coastal sea waters of Shieldaig on the west coast of Scotland in northwest Europe. Fish farms use 2-year production cycle for Atlantic salmon. After hatching in freshwater tanks, the young salmon was transferred to seawater cages. Atlantic salmon spend 2 years growing in the seawater cages before they are harvested. Infections by salmon lice on the farm salmon are known to be higher in the second year of the 2-year production cycle. So in the 2-year production, the second year is when they are more vulnerable. The scientists wanted to test the hypothesis. Infections by salmon lice on wild sea trout in coastal seawater at Shieldaig will be higher in the second year of the 2-year production cycle of the fish farm near Shieldaig. Electrofishing was used to sample wild sea trout population in coastal seawater regions near the fish farm. A boat was used to travel to the sample location and electric current was applied to the water around the boat. This caused some fish in the area to stop moving temporarily. A hand net was used to catch the fish that were not able to move. The number of sea trout caught was then recorded. The number of salmon lice attached to each sea trout was also recorded. The fish were then returned to the sea. Sampling was repeated once a year for 10 years. State one variable that should be standardized in this procedure other than the boat and the hand net. So, one thing that can come directly in our mind because we are using electric current. Alright, so we can talk about the electric current that needs to be, standardized. So first point, we can talk about the electric current applied needs to be standardized. Now, other points that we can write include the location of the sampling site and the time of the year. So any one of the options need to be written to get the 1 mark. Electrofishing is more likely to prevent movement of larger fish. State why this systematic error may give results that are not representative of the wild sea trout population. So, as we read from the data that it can only restrict the movement of larger fish means young or juvenile fish will escape. and they are not caught. So the answer can be written: small trout will not be caught, or there will be more lice on the fish that we caught because they are older. The scientists collected data on infections by salmon lice on wild sea trout in coastal sea waters at Sheldeck every year from 2000 to 2009. During this time, the fish farm near the Sheldeck completed their 2-year production cycles 5 times. The data are shown in table 2.1. So looking into the data, we can see between 2000 and 2001 there was one cycle, and on the second year of the production, percentage of the sea trout attached with salmon lice were higher. In 2003 it's still higher, but in 2004 and 2005 the percentage trout with salmon lice were 0. So alright, we can see sea trout. In the first one, the data for greater than 13 attached salmon lice were not collected. And in 2004-2005, there is no salmon lice that is attached to the sea trout. In 2007, 2006 and 2007, we can see percentage of sea trout attached with salmon lice has increased dramatically in the second year. And in 2009, it decreased back again, right? And also in the same 2007, the highest percentage of sea trout were attached with more than 13 salmon lice. Explain why the scientists calculate the percentages of sea trout with attached salmon lice. So the answer can be written: calculating the percentage allows for valid comparison between each year, and because the number of sea trout sample varies each year, the scientists then calculate the mean percentage of sea trout with attached salmon lice in the first and second years of the fish farm production cycle. This means are shown in the table 2.2. The year of production cycle, on the number 1 year the mean percentage is 61, and on year 2 the mean percentage is 33.2. So in the next one, greater than 13 in salmon lice in the first year is only 2.6%, whereas in second year is 14.9%. The scientists concluded that the greater the number of wild sea trout in coastal sea waters at Sheldeck are at a risk of death in the second year than in the first year of the 2-year production cycle of the fish farm near Sheldeck. Use table 2.2 to describe the evidence that support this conclusion. So the answer can be written in this way. In the year 2 production cycle, there is 12.3% greater sea trout attached to greater than 13 salmon lice. Now we already know from the experiment, if greater than 13 salmon lice is attached to the sea trout has a very high chance of death. Suggest limitations of the data shown in table 2.1 and 2.2 that reduce confidence in this conclusion. So the answer can be written as follows. There is no difference between the year 1 and year 2 of production cycle in 2004 and 2005 shown from table 2.1. The number of sea trout sampled was really small. Now this too can be achieving the full marks, or we can add other points. The data is from 5 production cycle only, so the data is limited, or there is no statistical test carried out to increase the confidence of the data. The scientists then decided to use published data to investigate the effect of distance from the fish farms on infection of wild sea trout by salmon lice in northwest Europe. 8 locations in coastal seawater regions of Scotland and 17 locations in coastal seawater regions of Ireland were sampled over several years. The number of salmon lice attached to each wild sea trout sampled was recorded. For each location, the distance from the nearest fish farm was recorded. The scientists predicted that the number of attached salmon lice on wild sea trout will decrease as the distance from the nearest fish farm increases. State a null hypothesis the scientist would make before carrying out the statistical test. So a null hypothesis, is made by saying no significant difference. No. So we can write down, there is no relationship or correlation between the number of lice on sea trout and the distance from the nearest fish farm. The scientists then carried out statistical tests on the data to see if the relationship between the number of attached salmon lice on wild sea trout and the distance from the nearest fish farm was significant. The probability values from the result of the statistical test are shown in table 2.3. In Ireland, probability values of 0.001. So significance, it will be definitely significant, yes. However, in Scotland, the probability is 0.559 values of P. We have to remember our exam accepted probability is 0.05, meaning we will accept 5% due to chance. And here the data shows almost 56% due to chance. So this data is not significant. The answer will be no. And this one, it shows less than 0.1% due to chance, which is definitely significant. Complete table 2.3 by indicating the results of the statistical text, whether they are significant or not significant. Figure 2.3 shows the distance from the nearest fish farm plotted against the mean number of attached salmon lice per wild sea trout. The error bars in figure 2.3 shows 95% confidence intervals. So the error bars are representing that how far away this particular average lies from. Greater error bar decreases confidence. An overlap of error bar indicates no significant difference. Now, the mean number of attached salmon lice per wild sea trout, distance from the fish farm in kilometer, as it increases, it decreases. In Ireland, it decreases much faster in comparison to in Scotland, which decreases, then slightly plateaus and then decreases back again, but the gradient is not as fast. With reference to figure 2.3, state the relationship between the distance from the nearest fish farm and the mean number of attached salmon lice per wild sea trout in the coastal seawater regions of Ireland. So the answer can be written, there is no negative correlation. We can say there is a negative correlation, or we can say as the distance from the nearest fish farm increases, mean number of lice per trout decreases. With reference to figure 2.3 suggests why the scientists calculate the 95% confidence intervals for their data. Scientists calculate 95% confidence intervals for their data because there is a 95% chance that the true mean lies within this range, and the narrower the error bars, for example for Ireland, the closer the calculated mean is likely to be for the true mean or the true average. Guys, that's all for this particular question paper. Thank you for watching this particular video. See you in the next video, guys, and best of luck for your exam preparation. Bye bye.